speaker, Dr. Paul Wischmeyer, who's a critical care, perioperative, and nutrition physician and professor with tenure in the Department of Anesthesiology and Surgery here at Duke. He's also the Associate Vice Chair for Clinical Research in the Department of Anesthesiology, directs the nutrition team at Duke Hospital, as well as the Perioperative Research Group here at the DCRI. So really excited to hear about this new emerging area of research and the projects and work that Paul is doing. Dr. Wischmeyer earned his medical degree with honors at the University of Chicago, where he was elected into the Alpha Omega Alpha. He's received numerous awards from national and international societies, including the John M. Kinney Award for the most significant contribution to the field of general nutrition research. He has over 140 peer-reviewed publications in nutrition. Index is 48. So please join me today in welcoming Dr. Paul Wischmeyer.
Thank you. Just going to restart this to be sure it runs. So we spend a lot of time in medicine trying to eradicate bacteria, trying to do away with all the evils that bacteria, germs, and all the other things we call them bring. But I guess one of the questions I want to pose to you is perhaps we shouldn't be. And perhaps we should be seeing the world a whole different way. I'll show my disclosures just briefly. I like to call them alignment of interest because I'd like to believe that the things that we want to do to improve people's health are the same things these people want to do to improve people's health. And so rather than conflicts, I like to think of them as alignments. So back to our question, though. Is it possible that instead of bacteria really being the enemy, that realistically these bacteria are really 100 trillion friends you didn't know you had, and perhaps you want to keep around? And so, in fact, to the point perhaps where maybe someday we'll start giving them back to you in different ways. Maybe even books you can read at home will teach you how to give them back to yourself in different ways, and someday maybe we'll be writing prescriptions for stool transplants, stool pills. The kids at MIT will sell you their stool in a pill form that you can take, and they'll tell you it makes you smarter. The FDA has some problems with that, but nonetheless, they'll sell it to you anyway. So, but this day may be coming. And this day has already come for some people. This young woman named Caitlin definitely came for her. She was a young woman who lived in Atlanta, and she underwent, unfortunately, a severe car accident. And she was in the ICU, much like the ICU I work at here at Duke, for a month. And she was getting better, and just before she was getting ready to go home, she started to have diarrhea. And she was diagnosed with Clostridium difficile, which is most believe the fastest growing iatrogenic cause of death we have in the hospital today. And she got 9 different rounds of antibiotics for this Clostridium difficile, to the point where she was dying of it. She was days from dying of it. It was killing her. And they decided to give her a stool transplant from her mother. And miraculously, the next day, she was better. And she stayed better and never needed antibiotics again. It saved her life. So undoubtedly for Caitlin, the 100 trillion friends she didn't know she had, she definitely needed. And so it brings up the question, when we do all the things we do in modern medical care, much less the antibiotics you give your kids at home or that you are taking right now for the flu you have, are we not eradicating what ultimately I will show you makes us human and perhaps makes us healthy? And is it possible we should be giving these back, whether it be with probiotics, stool pills, or stool transplants? Is it possible that this is the future of a big part of medicine? And so that's what we're going to talk about today, because it's key for you to remember that the world we live in is largely a microbial world. We as humans and multicellular organisms make up that very tiny little red area on the great tree of life. The rest of it is largely single cellular bacteria that make up most of the world. Why should you care about your microbes? Well, have you ever wondered why going to the woods you get bit by all the mosquitoes and your wife or husband or friends don't? It's not because you're sweeter. It's because the bacteria that live on your skin actually attract or repel bacteria with the chemicals, mosquitoes with the chemicals they secrete. In fact, whether Tylenol is toxic to you may be as much determined by the bacteria that live in your gut as it is the amount you take or how you metabolize it. There's a lot of data that shows actually your microbial makeup of your gut tells whether your heart failure drugs are going to work or whether Tylenol will ultimately injure or not injure your liver. In fact, the microbes that live in your gut may actually decide who you have sex with, if you're a fruit fly. We know without a doubt that the microbes that make up the guts of fruit flies clearly determine sexual preference and mating preferences. Maybe that's true for humans as well, and we just haven't figured it out. We'll see. So how do they do all that? How do they do all the things they do? So this comes back to what really makes us human. Perhaps you think it's our cells that make us human. Well, there's 30 trillion human cells in your body right now, and about 38 trillion microbial cells. So cellularly, you're definitely more microbial than human. So then a lot of people think it's our genes that make us human. Of course, there's 20,000 human genes in the body right now of all of you sitting in the audience, including myself. There's 20 million microbial genes living in you right now. So you are genetically 1% human and 99% microbial. So it's no wonder they determine so many things in our life. In fact, there are about as many E. coli in your gut as there are humans on the earth. And that's right now. And despite what we may think, all humans are 99% identical, as different as they may seem, genetically. But the microbes that live in your gut right now, the 2 E. coli sitting next to each other in your gut, maybe have genomes that are perhaps 40% related. They can be significantly different. So there's an incredible amount of diversity, not so much in humans, but definitely in bacteria. So sadly, despite what your mother told you, not so much a beautiful snowflake and unique, but your microbial symbiotes, they are. They are, without a doubt. So what's exciting now is we're poised to answer key questions about our microbial selves and about how they can be manipulated to change health. The number of publications for the microbiome is skyrocketing. This is only data up to 2012. The graph wouldn't stay on the screen if I went further than that. So this is clearly an evolving field. Why is that? Well, the reality is until very recently, we could only culture or see under a microscope about 5% of the microbes that exist on Earth, or in our bodies for that matter. But now we can see all of them through sequencing. This is where the word microbiome comes from, this microbiome sequencing that we can do. And interestingly, when we sequence all this data, we have to show it to you somehow. And so one of the ways to think about it is we're going to show it to you as a map. And of course, if you look at a map, all the organisms, flora and fauna, that make up a particular area of the world are different from one area to another. And so much like the different organisms and animals and plants that live in different parts of the world are different, the bacteria that live in different parts of you, whether it be your mouth or other places, your gut, are different. And so when we do all this analysis of the microbiome, we generate lots of ATCs and Gs. In fact, the US government spent $174 million analyzing 250 healthy volunteers' microbiome. I want you to remember that number, because I'm going to tell you about another project that made some money doing this in a minute. But it generates a ton of data like this. Not very exciting. It's a lot of money to generate that. But how can we map this? Again, I want to emphasize that different parts of your body have very different microbial makeups. So when we map them, we get a map that looks like this. And of course, all bacteria look about the same under the microscope, and they look about the same on the screen as well. But when you plot the differences in them, much like on a map, you get different locations based on the different parts of the body that they come from. So if we sampled your mouth versus your skin versus your stool, we get very different results of what lives there. In fact, the difference between your mouth and your stool, just a few feet away, is greater than the difference between all the organisms on this coral reef and all the organisms on this prairie. So a few feet of distance in your body makes up a huge diversity difference, much greater than thousands of miles on Earth. In fact, even within two people's fecal content, you can get enormous differences, bigger than, say, the plains of the US or, say, the forests of Asia. So enormous differences even within individuals can occur. How stable is the microbiome? Well, these are actually plots of people who collected their microbiome every day for years. There are people that do this. I'll tell you about people who do this with their kids in a minute. But nonetheless, it's very stable, actually, in an individual person in all parts of the body. So unless we do something to perturb it, whether it be illness or iatrogenic intervention, it stays pretty stable. So where does our microbiome come from? And I have to tell you, if you have a dog at home, yours is different than someone who doesn't have a dog. So pets change your microbiome. But the reality is our microbiome comes from birth, right? And for thousands of years on Earth, all babies are born by vaginal delivery. And in fact, all of the microbiome of an infant for thousands of years on Earth, whether it be stool, mouth, or skin, resembled mom's vaginal flora. Until recently. Now, of course, babies are born not infrequently by C-section. Now, for the first 2 years of life, all of the microbiome of the infant resembles mom's skin, which is not how our bodies were built to tolerize, which is why we think perhaps there's greater risks of allergy and different other diseases in children who are born vaginally versus C-section. Now, my partner in crime, who I'll talk about in a moment, Rob Knight, who helped do the Human Microbiome Project, when his child was born by C-section, he kicked everyone out of the delivery room and smeared vaginal secretions from the mom all over the baby. And I would encourage all of you to do the same. It's probably the only thing you remember from this talk, but you'll do your infant a favor because that's what we have evolved over millions of years, or thousands of years, depending on what you believe, to tolerize to. And so that's key. So you've seen a child develop psychologically, you've seen a child develop physically. Here's a child developing microbiome-wise. This is bacteriologically. So this is someone who sampled their child every other day for the first 3 years of their life. Yes, people do this. And this is what it looks like. So this is the infant's fecal sample. So they sampled the baby's poop from the diaper every day, every other day, and you can see it starts off looking a lot like mom's vaginal flora. And then it begins to transition towards what you hope will be the fecal flora down here. But then something's going to happen. The child's going to get an ear infection. And this child's going to get antibiotics. This is 1 week of antibiotics. Reverses years, or many, many months, of evolution of the microbiome. So all of the gain, luckily this child recovered, but all of that gain that was made over months of development is lost in as much as just a week of antibiotic treatment. So you can see that again. It's a huge effect of even just a few days of a narrow spectrum antibiotic on a child's microbial makeup, or ours humans' microbial makeup. Luckily for this child, by day 838, the child's stool begins to look like an adult stool. And so this child eventually gets to where it needs to be, starting off looking much more like mom's vaginal secretions. Do changes in the microbiome matter? Well, of course, we think they do. We know, of course, that getting antibiotics as a child is associated with perhaps obesity, autoimmune disease, allergies, and other changes in ultimate health status down the line. In fact, we can predict with 90% accuracy, just looking at the stool from one of you or another human, who's obese and who's not. Not very useful, actually, probably. But if you looked at all the genes of that individual, you'd only be 60% accurate. So the stool microbiome is much more accurate than the genetic makeup, and probably, we believe, plays a bigger role. And so, can we use this to advance science? Can you transfer these traits, taking the stool from one individual and giving it to another? And so this has become a whole science unto itself. So you can take a mouse that's been raised in a germ-free incubator, we call them germ-free mice, notobotic mice, and you can give them stool from another animal and transfer traits. So in fact, you can take the stool from an obese mouse or rat, give it to a germ-free mouse, and that mouse will get obese. And it's not just because it changes metabolism. They actually eat more. There's a whole science. In fact, the NIH's first funding call for microbiome was on gut-brain interactions. And now what happens in your gut changes what happens in your brain dramatically. In fact, then if you take the stool from a lean or normal mouse and give it to the germ-free mice, they stay lean. And so you can transfer traits. You can take stool from an obese human, give it to a germ-free mouse. The mouse will get obese. So you can now transfer human traits and human, perhaps, disease and recreate it just by transferring the microbiome into the mouse. So, in fact, in a really fascinating study done in Malawi that Rob Knight helped work on, they took twins. That one twin, as it turned out, was starved, had kwashiorkor. One twin was well-nourished. And they took the stool from the starved twin and gave it to the mouse, to these germ-free mice. The mice got cachectic, lost weight, became starved themselves. So what's in our stool, what's in our microbiome fundamentally makes up in so many ways who we are, and in fact could possibly be used to change even worldwide hunger and starvation and some of the diseases that plague all of the world in such a big way. So can we more meaningfully use it to cure disease in the clinical setting? Clearly, we've done this before for people like Caitlin. So there is that potential. And so this is one of those questions that someone like me who's interested in surgery and critical care and acute illness in patients have become very curious about, because all the things we do to patients in the ICU and in the operating room and other parts of care affect the microbiome. Our own endogenous catecholamines change the way our bacteria see us. All of us have Pseudomonas that sit in our gut right now, sitting quiescently, waiting for the hurricane to come or the stress to occur. And it senses that by sensing our own endogenous catecholamines, opiates, the administration of exogenous or endogenous opiates, changes the virulence of the microbiome, changes the aggressiveness of those Pseudomonas sitting in your gut right now, tells them to attack. And so all these different things, and of course antibiotics themselves are the biggest offenders by disrupting the community. And so the question is, when we treat with all of our advanced antibiotics and all of our advanced drugs, do we need to restore balance? Probiotics, stool pills, other methods of restoring what was there before. Again, should we resod the lawn that we disrupt? One way to do that, of course, is probiotics. And they're everywhere, right? They're in half of the things we eat, or at least people would like them to be. And there's books about them that can tell you all about what they do for you or maybe what they don't do for you. And so probiotics, perhaps, are becoming a very useful future for us. And in fact, there was a recent trial in Nature that showed that giving a probiotic to well-term infants, 4,000 of them in India, actually reduced all-cause infection in those infants when they were followed for long term. And so, again, it's the biggest thing to hit the probiotic literature ever. It's a large NIH-funded, actually was done out of Nebraska, investigator over 10 years, who showed that you can actually change populations' health by giving probiotics. But what's the science behind them? What do we think they do? So coming back to what we talked about before, we know that, again, the organisms that live in our gut sense our stress. I trained under John Alverdi and Eugene Chang at University of Chicago. John Alverdi, actually after I was in the lab, later in his career about the age I am now, discovered quorum sensing and discovered the bacteria actually talk to each other when we become stressed and they attack us. And one of the ways they sense that is clearly all the changes I talked about from the drugs we give and the stress that our body shows. But when we become sick, I will show you, and we found out that the normal bacteria that live there go away. And it allows the pathogens to predominate. It's a little bit like in a hurricane, right? People don't loot stores when everyone's around the city, say in New Orleans, but then the hurricane comes and everyone leaves, people start looting and they bring their friends to loot too. That's what bacteria do in your gut. They wait around for the storm to happen and then they attack. And so we lose our commensal flora and we lose our ability to protect ourselves. And so we've seen this in ICUs and operating rooms where there's loss of these normal bacteria, at least in culture studies, and this may associate with bad outcomes. And so the idea would be if you put back all the people in the city, the looting would stop. And if you put back the commensal bacteria, the patient would recover, and they wouldn't have all the adverse complications, perhaps, that they have if those aren't there. But then, of course, there's the idea that giving bacteria to people may be risky. In fact, the HRQ was sure it was risky. They spent a whole bunch of money a few years ago putting out a 650-page document, sure they would find harm from giving probiotics. They looked at 622 trials, some going for years long, looking for such signals of harm. They didn't find any. This is a meta-analysis table, forest plot of all the different risks that could be found in well and sick people, and again, the confidence or the relative risk is right on 1. So there were no risks. And in fact, if you look at the sick populations, this is probiotic use in acute illness, there was even a trend towards benefit, less adverse effects in those patients. And then there was actually one trial, which is useful for those of us who think about clinical trials, which is probably everyone in this room, that did show harm. It was an interesting trial. It was to treat pancreatitis with a fiber and new 9-species probiotic mixture in the Netherlands. And there was a lot of ethical concerns expressed around this trial. So at the first interim analysis, they noticed there was one group that had a fair bit more mortality than the other. And they thought, oh, the probiotics are working. So they broke their stopping rules and kept going, hoping to get a bigger signal. It wasn't that group. What was happening, pancreatitis, for those of you who ever cared for someone with that, leads to severe ileus. And they gave these probiotics into the small bowel, not into the stomach. where they could sit and not move, and with that wonderful fiber bolus grow sitting in one spot. I talked to the surgeons who operated on these patients. They'd find these balls of fiber and necrotic bowel right around it because it didn't move. And so, again, all kinds of issues came to this, but we learned you never give probiotics postpylorically, and you don't break your stopping rules. So there's a reason, and this was a shock, and it got a lot of press because of it. This was the only trial that's ever shown harm in any probiotic trial out of 622. But it's an interesting case study in what happens when you don't follow your rules. So they concluded there is no risk, adverse risk, at least in all the human trials done to date, of giving probiotics or taking them at home or taking them in the hospital. And so significant safety. So do they improve clinical outcome? What's the data? Well, some of the most profound data was published in JAMA recently, about 12,000 patients looking at the use of probiotics to prevent antibiotic-associated diarrhea. Have you ever taken an antibiotic and gotten diarrhea? A lot of people get that. Nobody wants to admit it, but a lot of people get that. And so they used a whole slew of different kinds to treat that, and they found a 40% reduction in antibiotic-associated diarrhea when a probiotic was given. It didn't seem to matter which one. Again, the bad actors that cause diarrhea are slow growers who don't want to deal with the rest of the city being populated. So once you repopulate it, C. diff is included in this, once you repopulate it with normal bacteria, no matter what they are, the bad actors go away. And this is what they found in this trial as well. This is a trial of preventing ventilator-associated pneumonia, which is a big problem in our ICUs. We don't get paid for it anymore, so we don't report it very well because we want to not get paid. But the reality is a fair percentage of patients still get this around the world. Giving a probiotic in an NIH-funded trial, Lactobacillus GG, actually the culture oil you can buy at the store, they're not paying me to tell you that, reduced ventilator-associated pneumonia by half, 50% in the intention to treat group as well. And so this was actually a friend of mine's K grant, who did his K23 project on this and got a nice publication out of it. And so this is one of the more compelling studies, well-done studies showing the benefits. And then my group has done meta-analysis work on this for years, and our most recent found 30 trials. These are in ICU patients, our sickest patients, looking at probiotics of different sorts on infections. We found significant reductions in infections, reductions in ventilator-associated pneumonia, and the sicker you were, the more likely it was to help you. Again, the sicker you are, the more deranged your gut is, our hypothesis was. And that data proved to be true. So this is the plot for that. Again, you can see about a 20% reduction in the data we have now. So again, there's some challenges. We still need better trials. There's a lot of different probiotics being used. There's a lot of heterogeneity in the trials. And there could be publication bias, some of our plots showed. So a lot more needs to be done here. So this is true in other areas, elective surgery, sepsis, and other conditions as well. There seems to be positive findings in every field that we look at for the potential benefits of probiotics on outcome. But we need better trials. Of course, one of the most pressing issues we face as a country, much less the world, is C. diff. When I was a resident, which wasn't that long ago, I saw a case of C. diff every year maybe, and nobody died of it. Now it's a rapidly growing cause of death, the last New England Journal paper would say, and hundreds of thousands of people have it every year. In fact, I had a patient a few weeks ago here at Duke who had a small nasal surgery, had some bleeding in my ICU for the bleeding. Before he went home the next day, he told me he'd had diarrhea for a week. He'd only gotten 2 days of Periop antibiotics, narrow spectrum. I'm like, no way he has C. diff, but I should check him. He already went home. By the time we got the result, he was positive. So even 2 days of perioperative antibiotics can cause this devastating disease. Is it possible we can cure it using either the patient's or someone else's stool? This has been published, or probiotics itself. This probiotic data has been reviewed in the Cochrane analysis, 4,000 patients, many trials, 60% reduction in C. diff just by giving a probiotic of different sorts to these patients. So very effective. Again, C. diff is a weak little organism that in the face of other organisms won't grow. But we have great antibiotics that wipe all those other organisms out, and I'll show you that in a minute. Worked in kids and adults, worked at different doses, worked with different species. Again, the fast growers are always going to grow faster than the C. diff. And we've already shown it could save her life, but we saved her life not with probiotics, but with stool transplants, which have a more than 90% cure rate when they're given around the world for this condition. What happens to the microbiome, actually, if you measure it when you do a stool transplant? People at the University of Minnesota have done that. Again, this is the healthy human microbiome population that I showed you before. These stars represent patients who had C. diff. These are stool samples. They're supposed to be down here. They're not. They're shifting up towards the mouth and the skin. Remember this curve, you're going to see it again. It's a little troubling. So they took a number of these patients and they gave them a donor stool transplant, and watch what happened to their microbiome as you followed it over a few days. It immediately went back to the normal fecal constituency of the donor and stayed there. They continued to sample this patient for weeks. So this was an incredibly potent observation showing the effect of a fecal transplant on recovery of the abnormal microbiome of the critically ill patient with C. diff. So clearly we can restore balance, and we can make these patients better. So where do we go from here? Well, my first thought, and those I work with in the critical care trials groups, large clinical trials are probiotics. Maybe we should stool transplant everyone. Maybe all of you should be freezing your stool with the idea you might get hit by a bus. In fact, I would probably do that, but nonetheless, or we should all be taking poop pills. Of course, even the TV doctors know it's not that simple, right? So we went to the NIH with a big trial, and they said, no way. One size doesn't fit all, because we went with Lactobacillus GG, a single probiotic. We were going to treat everybody, right? And they didn't buy it. Well, we should have known that. I can tell you, for those of you who don't work in the critical care space, we've done now 80 major trials of more than 1,000 patients in the ICU of an intervention. 72 have been negative. So the phase 2 was positive, phase 3 was negative. 6, actually, phase 3 hurt the patient. There was this one drug called Xigris that cost $20,000 a dose and made you bleed from your eyes. It was a coagulant drug that worked for a while. And then 2 more trials showed it didn't. It had too many risks, was too expensive, got taken off the market. So we've never had a positive trial of more than 1,000 patients of a single intervention in a broad population. So they said, define the microbiome in the patients first and tell us what's missing. And then come back to us with a trial that actually targets people with what they need. We don't buy that one size fits all. And then you can tell us what's supposed to be in these poop pills or whatever other thing you're going to give the patients. So a group I work with, we decided to do this a few years ago after getting summarily rejected in our proposal letter. And we created the ICU Microbiome Project. We used an existing survey database that we had built. It surveys thousands of patients around the world and hundreds of ICUs. We had dietitians, nurses, and pharmacists collect all of this data for free. Everything I'm showing you now was done because people cared. And we collected microbiome stool samples, oral samples, and skin samples across a number of ICUs around the US and Canada. And we thought, gosh, if we can see what's missing, we can guide therapy, maybe we can guide diagnosis, predict risk. There's a lot of things perhaps we can learn from doing this in our patients. So this is our first publication, came out about a year ago. Looked at our first 115 patients from 4 centers. All these people were quite sick. They'd all been in the ICU more than 48 hours. They'd all been on a ventilator more than 48 hours. And 100% of people, if you're in an ICU in the United States for more than 48 hours, will get an antibiotic. We couldn't find anybody that didn't, which is interesting. They were pretty sick. For those of you who know about Apache scores, that's how we standardize sickness. All got antibiotics, fairly long length of stay, almost a month in the hospital, reasonably high hospital mortality. These were pretty sick people. We compared them to a 1,242 patient, well, healthy individual control group from what's called the American Gut Project. Actually, we just resubmitted it to Science for the second time last week, fingers crossed. And so these were all healthy people who hadn't gotten antibiotics recently, didn't have IBD or other chronic diseases. And this is that group. This is the group Rob Knight, the scientist I told you about, has created. He's now studied 6,000 people. And what he does is he has people send them their stool, or their wife's stool, or their dog's stool. Pay him about $100, he'll send you back your microbiome profile. I can't tell you what to do with that, but people do it anyway. He's raised $2 million analyzing 6,000 patients' microbiome. It cost the NIH $173 million to analyze 250 people. And he's made $2 million analyzing 6,000. So that's the other way we paid for this trial. So it's pretty amazing. He's the largest crowd-funded scientist in history. At least we think he is. So our hypothesis was this, that a healthy gut is a diverse gut. And that being sick, being in the ICU would lead to loss of diversity. Makes sense, and that would associate with bad outcome. So these are results, only the most significant ones, because we did a lot of comparisons, had to make a lot of corrections statistically. And there was a clear shift from normal microbiome to what we call pathogens or proteobacteria in patients in the ICU. There's those plots again. These red dots are the healthy people. These blue dots are the ICU patients. There's a significant shift in what their stool looks like. There's a significant shift in what their mouth looks like. Again, the same. This is all of them together. Again, this is the normal human microbiome project. There's a nice spacing here. Remember I told you that the gut and the mouth should not look the same. They begin to look very much the same. The big dots are the ICU patients. So our mouth and our stool begins to look quite a bit the same as we lose our health and become quite sicker. We believe this is barrier function, but really loss of diversity. In fact, if you look at specific bacterial species, looking at that plot to this plot, you can see there's almost a complete loss, the white dots being very low numbers of a normal commensal firmicute family versus very high amounts in the normal individual. So whole families of bacteria are lost within 2 days in the ICU and perhaps in other illness states. And another way to look at this is to look at all the different families in these kinds of plots. The red up there is all the pathogens. This is a normal plot of each one of these little lines represents a different subject. And so there's normally not much pathogen-related bacteria in our guts. In the ICU, there's predominance of pathogen-related bacteria in our guts, so you can see. And it gets worse as the longer you stay. That's admission sample and discharge sample there. And so this marked increase in pathogens occurs. There's some specific bacteria that are depleted. One in particular that we found is one of our targets, this drug called Faecalibacterium, or this bug called Faecalibacterium prausnitzii. Makes short-chain fatty acids in our intestine, which is the main fuel for our intestine. It's depleted in IBD as well. It was dramatically depleted consistently, and now other ICU trials in kids and other populations have shown this is depleted as well. This may be one of those targets that NIH was looking for. And then there's enrichment of all kinds of pathogens in the stool, but some of the common ones you'd see in hospital care, things like staph and proteus, grow up very richly when we become ill. Same in the mouth. Mycoplasma seems to grow up richly, a few other staph as well in the people who are sick, and there's loss of normal oral flora as well. In fact, you can see there's this dramatic increase in the ICU patients of known pathogens, things we culture in the blood of our patients in the hospital all the time, and there's loss of important protective bacteria that perhaps we should be giving back. Diversity was, again, one of our questions. This is black lines here. This is a diversity plot, so the further you go this way, the more diverse you are in your bacterial flora. I can tell you this is a Western diet. This is from a science paper that looked at a population on an island in the middle of the Pacific that had never seen Western diet. They were up like this, so this is not a healthy diet we eat every day. But the ICU patients are much, much worse. They have a much, much lower diversity than even a Western diet, sort of semi-healthy diet. In fact, we were struck by the fact that patients could lose so much diversity that 95% of all the organisms in their intestine within a few days could be made up of one family, one taxa. So this is a patient that the most abundant species in its entire gut was 25% of all the bacteria. A few days later, one species was 95% of all the bacteria. It was an Acinetobacter, or an Enterococcus. It's almost always a gram-negative pathogen. So there's this crash in microbiome diversity that happens very rapidly in our patients that we're only now beginning to understand. We then began to look at the sources of what patients' fecal and oral samples look like. So we know, for instance, that what it should look like when you get a stool sample is these red samples here, which are from the healthy American gut patients. This purple that you see showing up a lot is pediatric. These are samples from children, babies and children. It's called the global gut population. It's a microbiome population. So our guts begin to look like a baby's gut. Very quickly in the ICU, it begins to revert back to where we started off as children. Unfortunately, in the mouth, our mouth flora begins to look this green color, much like the skin. So our skin and our mouth begin to look very similar. Unfortunately, we also begin to see, we have source samples from decomposing autopsy sites from crime scenes, dead people that have been there for a long time. People's guts begin to resemble that as well, which is even perhaps more troubling. So there's some quite disturbing pathogens that show up. So we've begun to do metabolomics of the fecal stream as well. And we've discovered some interesting things. Of course, metabolomics looks at the chemical compounds or drugs that show up in the fecal stream. We've done this in the blood for years. But we found that, of course, ICU patients have very different fecal appearance, metabolomically, than well patients do. And in fact, this correlates with scores of severity. You can actually correlate the change in how different it looks with the Apache score, which is a common severity of illness score, and with the Nutrix score, which is a nutritional risk score. In fact, you can identify specific compounds, like all the antibiotics, beta blockers, and other drugs show up in the fecal stream. We can see those. But perhaps more interestingly, we found something that had never been published in a human before, but had been seen in animals, and it was bacterial antibiotics. Bacteria make Pseudomonas make quinolone-like products. They look like fluoroquinolones, look like an antibiotic like Cipro. They do it to kill everything else off around them so they can predominate when they see stress in the body. We detected that in quite a number of patients. And in fact, the patients who got bacteremic, got C. diff, or got both had a much higher rate of this particular quinolone-like protein. So we can actually sense or see when patients are becoming stressed enough that their bacteria are becoming virulent. And we perhaps could make a very simple test someday that we could look at the stool of a patient and predict who's at risk for the complications of the ICU, looking at these Pseudomonas quinolone-like signals and other antibiotic signals. Again, we don't know if this has ever been published in a human. We plan to publish it soon. So clinical results as we finish. So interestingly, death was most significantly correlated with the amount of Klebsiella on your skin. So believe it or not, the bacteria that live on your skin seem to play some role in life or death. And there were a few other changes as well, which was surprising to us. When someone gets bacteria in their blood was predicted by mycoplasma, the thing that causes infection in your kids and sometimes in adults, showing up in the mouth. ARDS, acute respiratory distress syndrome in the lungs, so severe lung injury, had bacteria. I have no idea what those two do. We've discovered some relationships with even species we don't have a full understanding of. And in the stool, that was true as well. And then the effects of how we give nutrition change the mouth and the stool as well. And whether or not we gave IV or enteral nutrition did as well. Antibiotics, of course, became the big question the reviewers asked. Is all you're seeing all the antibiotics you're giving, or is it actually the illness, the ICU illness itself? And so we created an antibiotic pressure score from a Lancet publication, looking at all the antibiotics the patients got for different days and how long they got them, and tried to quantify that question. And we found actually in the mouth, the antibiotic pressure score did correlate highly with the loss of diversity in the mouth. So the antibiotics you got did drive what the mouth looked like, but it didn't in the stool. The antibiotics pressure that you got throughout your ICU stay or throughout your hospital stay did not drive what happened to your stool microbiome. It changed independently of that. And so that was not driven by antibiotics, but perhaps by the stress of the illness itself. And so it's more than just the antibiotics. Diversity, of course, predicted a number of things, but length of stay was a big one. Patients that stayed longer, that was correlated with a much less diverse microbiome. Chicken or the egg isn't clear yet. And of course, people with more diversity seem to stay shorter. But the real goal of this was to predict targets that we could give back to patients to treat them. And so we found a lot of similar targets that the IBD literature has found, where things that change in IBD that have become targets are changing in the ICU. And the Faecalibacterium prausnitzii I mentioned before has become a big one. We just got a grant across the street with the person who runs my lab, Jim Davidson. We obtained it in the John Rawls lab for the microbiome people that may be here. We got a grant to study that, to see if we can, in animal models of critical illness, give back this human bacteria that we found to be deficient, to see if we can change and ultimately make a drug out of that. And so the magnitude of the emission of diversity and change seemed to correlate with the ultimate outcome of the patient. The good news is this research is really easy to do. We got waivers of consent because it's literally waste material we're collecting on little swabs. So it's very easy to do this. And there's lots of questions still that we could ask. We have a huge data set just from the few hundred patients we have already. If you guys have questions, we'd love to have you dive into the data set. I can tell you that the people that do this, the person I work with, is a computer scientist because, again, this is omics data, and so you have to have some skill set, but I'm learning. Even the medical students here at Duke spend their weekends learning programming languages and statistical languages. So maybe people in the audience have the ability to do this. I don't. But luckily other people do. We have some cool ongoing projects. We just finished a collection with the Everest team, the Extreme Everest team. We collected microbiome samples from European climbers and Sherpas all the way up and down the mountain to see the effects of hypoxia on the microbiome. So we have stool and oral samples for actually a few months while they summited the mountain. Didn't. Some did, some didn't. But we're about to start analysis on this right now. And then today, actually, or yesterday, was the kickoff, although we've been rolling for a few months now, of the 1,000-patient surgery project here at Duke. For those who aren't familiar, the Duke Surgery Chair, Alan Kirk, has invested millions of dollars to characterize the surgical response to injury, microbiome, metabolome, genome, immune markers, clinical outcomes in 1,000 patients in 12 different surgical subtypes. We're getting an extensive microbiome collection out of this. And so we're going to get from a month before surgery to month after surgery of this. It's never been done before. We have nothing like that to compare. So kudos to the Duke Surgery Chair for investing in what will be a tremendous project, because we'll not only be able to get the microbiome data, we'll be able to correlate that to all the other omics data, all the other immune data, which is really, truly amazing. So in closing, what have we learned so far? Clearly, change in specific species of the microbiome and specific species of bacteria can lead to complications that can lead to death, in fact, and correlate with outcome of our patients. And a healthy gut is a diverse gut. And that's what all of us should strive for in our daily life and in our patients' lives. And loss of diversity, whether it be in the ICU or in other settings, could be IBD, could be a lot of different settings actually, associated with the poor outcome. And so this is something we can intervene on. In fact, perhaps we can change the world here, the microbial world that makes up who we are. Clearly, we have saved lives already doing this. And I'll also tell you that at Sloan Kettering, there's an NIH trial where they're stool banking people's stool before their bone marrow transplant, freezing it, and they're giving it back to them after their bone marrow transplant. So people are already doing this, so everyone get a bigger freezer. You will want to do this. And there's going to be books and ways to do this as well, but this is already happening in trials around the world. And so perhaps we should resod the lawn, whether it be with our own stool or someone else's, or probiotics or MIT poop pills, to give back the 100 trillion friends that we really need to optimally survive and thrive as humans, to restore the balance that we disrupt when we intervene and treat people who are sick, whether it be antibiotics for your child's ear infection or what we do in the ICU. And of course, with the new technology in the microbiome, we can make it so easy a child can do it. At least hopefully we can. If you are interested in your own microbiome, this is Daniel who did all the analysis for the ICU microbiome project I just showed you. He works in Rob's lab. You can send your own sample, or friend's sample, or sponsor a Child from Africa sample into Rob, and he'll send you back your data as well. Of course, we bring kids, at least when they were in Colorado, we did, into the lab to show them how the robots analyze the poop. Kids not so excited about that, but we think it's pretty exciting. And I have to thank Rob and Daniel. This is Rob sampling a Komodo dragon at the Denver Zoo before both of us moved out of Denver. And they helped a lot with this project as well, of course. And I'll be happy to take your questions. Thanks.
When you mentioned transplanting the stool from one person to another,
yes.
How is it done?
Sure. So it's done a couple different ways. There's some ongoing projects. Actually, WashU is leading up a project in twin obesity, where they're taking the stool from a pair of twins, the stool from the lean twin, and processing it and giving it to the healthy or the obese twin. And so there's two ways to do this. You can actually give it through a feeding tube or an NG tube into the stomach, more preferably beyond the stomach. Or traditionally for C. diff, it's been done by giving it through either colonoscopy or through rectal administration. And so it can be given from the top or the bottom. Both seem to be effective. The stool pills, obviously, you take by mouth. The probiotics, you take by mouth. You want to smear one in your mouth, actually, if you're in the ICU, we found, but because the oral flora is important, too. In Asia, I've been reviewing papers in the ICU with chronically critically ill patients where they're creating common donors, where they take 50 people who they've tested for other illnesses, and they create a pooled stool donor from the community to create diversity. Of course, the idea would be someone from your house or your own, assuming you were a healthy stool, would probably be ideal. So there's about a 5% autoimmunity rate, autoimmune disease occurrence rate, in stool transplant patients. And so there is apparently some risk to getting stool from someone else, and we don't know why that is with this small autoimmunity risk. And so a lot of people's thoughts is getting your own stool back may be the best, or these pooled donors that are occurring in Asia. So a couple different ways.
Dr. Wischmeyer, great talk. Thank you so much for sharing today.
Sure.
Question, please, regarding Crohn's disease and your phrase, what happens in the gut happens in the brain.
Yep.
For people who have Crohn's disease, have you done any studies on effects of depression or ways to improve their mental health while they're dealing with that condition?
So that research is really, literally in its infancy. There's animal research that shows you can change cognition. There's research going on even at UNC where they're looking at infants' development, cognitive development, and the microbiome makeup. And then the NIH has put out all kinds of requests for studies looking at depression, cognitive function, and other complications of aging and development with the microbiome as well. But the human data, the Singapore Medical School wants to put on a seminar on this in May and was asking me for speakers and names. And I reviewed this literature about 3 weeks ago, and there's very little actual hard human data to really be able to establish it. But I can tell you there's, because I'm pondering a grant going in myself, 50 to 100 review articles on the subject that try to link different things together, but there isn't a lot of clinical outcome data yet. But there seems to be a connection. I can tell the animals that you transplant the stool into definitely eat more and act differently. So it's interesting, behavioral changes definitely occur. So, yep. I can hear you. I can repeat it.
So thanks.
Yeah.
Wonderful, provocative
Yeah,
talk.
of course.
Sort of a broad question. So
Yeah.
it seems like the maturity of evidence varies depending on the therapeutic area and clinical application.
For sure.
I'm wondering to what extent some of the work that you're doing, particularly in areas with potentially more maturity, like the ventilator-associated pneumonia, have been incorporated into clinical practice guidelines, and if not, how far we are from that happening, what else we need to get there?
So the implementation has been challenging, as you might imagine, and it's very much institution by institution. So at the University of Colorado, where I came from, I couldn't get this through my P&T to save my life because none of these probiotic preparations are FDA approved yet. They don't have the GMP that a drug would have. And the NIH themselves has put a hard stop on research that doesn't use IND'd GMP, and there's only 2 of them that I know of, maybe a few more now, but there's very few preparations. So it's really slowed the kind of research that we're talking about in, say, the Crohn's disease patient, which is unfortunate. Here at Duke, there's a couple probiotic preparations I can give. One of them is lactobacillus GG that you saw that trial described. And so there are places in the world that I visit that use them every day in every patient. And in Colorado, even though my liver transplant ID physician was the one driving the push to get them, we couldn't get them approved. And some of that's because we just don't have perhaps the validity of drug manufacturing that we have with other drugs. And so are they a drug? Are they a nutrient? Are they something else? And what do you do with stool pills? And what do you do with fecal transplant? That's a world to itself. So it's a challenging implementation, for sure. Yep.
I think you spoke to this a little bit, but is there some of this that can be handled with simple nutrition, both at home and in the ICU?
We do think so. So one of the plagues, of course, that we face in the ICU, in fact, the grant I'm writing right now that goes in on Thursday, looks at the U.S. as the poorest feeding nation in the world in our sickest patients. So if you took one of these ICU patients like the trial I showed you, we can show from our surveys for 15 years now that they get less than 50% of the protein and the calories they need for not a few days, but 2 weeks after ICU admission. And so we are really good, and it takes, on average, about 2.5 days to start any nutrition at all in a patient who's that sick. So we're really good at starving people, which I can tell you 1 day of starvation significantly changes the gut microbiome. And what you eat, of course, significantly changes the gut microbiome. One of the trials we're doing across the street is a prebiotic fiber to try to drive the nutrient source for the commensal bacteria. So there's these fructooligosaccharides that actually a lot of the formula companies are putting into their formulas, and the dietitians don't think there's any data to them, and there isn't. And so we're trying to see, do these fibers actually help the microbiome or not? So we're measuring microbiome samples every other day on people getting these versus a non-fiber containing formula. So there is a possibility. One caveat on that, so I speak on ICU nutrition in India, and we talk a lot about delivering high protein. And if you're someone from India, you'd say, well, I'm a vegetarian. Our patients don't take in very much protein. Well, as it turns out, 20% or maybe even 40% of the essential amino acids your body makes can actually be made by bacterial fermentation of carbohydrate. And we think in countries that largely eat less meat, less protein sources that are not anything but vegetarian-based, they're quite good at making amino acids. Of course, as soon as I give you antibiotics for 2 days in my ICU, I wipe all those out. And so your ability to do that is gone. And so we do think that there's a large role nutrition plays and how that interaction with the microbiome occurs, especially in disease recovery. Yep. Please.
slides, and that's the urine.
Yes, so I can, yep, it's not. It's not.
So I was curious as to what we know about that, and are there links between, for example, what do we know about the gut microbiome and catheter-associated UTI in the ICU, or urinary tract infection in the general population, which is both huge, very common incidents and very expensive and potentially harmful.
No, great question. So as part of this 1,000-patient project, Todd Purvis is helping lead the urine microbiome portion of it. And in fact, there are sites around the US actually very interested in those questions who are perfecting. I can tell you the best way to collect a microbiome sample is an art unto itself. We have a whole committee here at Duke that we've built of us that are interested in this to optimize this. There's a company actually that's made a solution that, because for the human microbiome, this projects that you saw here, people would just take swabs of stool off their toilet paper and send them in. Of course, the problem with that was there was these blooms that would occur sometimes if the sample didn't desiccate, and you'd get all these pathogens growing that they knew weren't real. And so you have all these filters, and that's what the science paper revisions are all about, actually. They don't buy our bloom filters necessarily. In the ICU, it's even more of a problem. So they've created solutions, companies have. They've seen an opportunity where you can preserve instantly, once you drop it into the solution, the microbiome content and nothing changes. And it's good for 2 months at room temperature. They're using them in Africa, of course, to do all these trials in Africa. And now we're using them in this surgical project. And so we're looking at urine as well. And luckily, some of these new techniques of collection of the microbiome that keeps it stable and preserved allow us to do things we couldn't do before. And so the urine will be fascinating. And then tying, say in the 1,000-patient project that we'll have probably a fair bit of UTIs, urology is one of the groups actually we're studying. It'll be interesting to see. I mean, of course, the stone people are interested in it too, and what relationship the microbiome has to stones. And people think there's something, but a lot of work to be done. It's a great question.
When you provide antibiotics to the patients, you indicated that it just will kill the normal flora in the gut.
Yes.
And I've heard, and I don't know how true it is, that sometimes they really don't come back to what they were afterwards. What do you think about that? And in that case, what is the long-term effect of using the antibiotics?
So there's Dr. Turnbull at Stanford has done studies like this. He did a fascinating study where he took healthy volunteers and he gave them 2 courses of ciprofloxacin, and about 6 weeks apart. And they couldn't have taken antibiotics before within the last year. And what they found was some patients who got sort of a routine antibiotic would have derangement and then recover very quickly. Some patients would have derangement and not recover at all, and the changes out to a year were permanent. Some patients that changed the first time they got the Cipro and then returned to normal, the second time they got it, changed and didn't return to normal at all. And so clearly permanent changes, perhaps for a lifetime, can occur from antibiotic dose or not. You saw that child recover in a week, but the Stanford data says some people don't. And the next time you take antibiotics, if you recover the first time, you might not. So it's not even maybe genetic or person to person, but it's event to event. And so there isn't a lot of data, but that is the seminal study that's been done on that topic. And there's more being done, but the recovery is really difficult. And so the idea is maybe we have to give things back to create recovery, because some people don't recover on their own. And how do you know who those people are? And it's a great question we don't know the answer to. Yeah.
You see a lot of peanut allergies now. You see a lot of different things. It seems like we see more and more of food allergies emerging.
For sure.
So I was just curious. I have a wife who has a lot of dietary restrictions, so that would be interesting to me to see what implications this would have on that.
Yeah, and if you talk to Rob or others that have spent their life in the microbiome field, they'll say that this change in C-section versus vaginal delivery plays a role in that, because what we are used to tolerizing to, we're no longer tolerizing to. And then the antibiotic use in the first 6 months of life, especially in the first 6 months of life, seems to have a fairly significant signal to these downstream allergies, autoimmunities, and other kinds of things that we didn't used to see before, but now we see all the time. And so we do think, and obesity maybe being the biggest, no pun intended, problem of all this, that we may be driving with some of this, and would there be ways, even in infancy, to look at this. The diabetes cohort in the world, so the diabetes researchers have got a large cohort out of Scandinavia where they've looked at kids with multiple sampling that is very intense early in life and continues through teenage years of the offspring of type 1 diabetics. is they have a long and rich sample cohort of frequent sampling that may begin to yield some light because it's big enough that you might begin to see some of these kinds of things happen and actually have the microbiome data to look behind it and discover some of what's behind that. There's some other sample cohorts too, but they have one of the very, very best. And it's all targeted at type 1 diabetes. Thank you. Thanks, everyone. And